Thank you very much for the nice introduction, and thanks for inviting me. It's a great pleasure to be here. So yes, I would like to talk about how to integrate different data types that are obtained from the same tumor and how to use networks. At least that will be most of my presentation. So the starting point here is I want to make a few remarks about the molecular tumor board in Zurich that we, together with other people in Zurich, have built up, have established in the last month or few years by now, where the basic idea, as most of you will know, and similar to the previous talk actually before the break, is to use molecular profiling, DNA and RNA sequencing mainly, in order to obtain a picture of the tumor, molecular profile of the tumor. And then, you know, from the raw sequencing data, there's basically a long way to go to really inform a treatment decision. And I will, of course, not talk about all of this, but certainly in the beginning, you have to process the raw data, quality control, mapping, and then in particular mutation calling. And then you have to interpret these mutations. You have to make sense of what the alterations mean. And hopefully, you also would like to predict what kind of action you can take based on these mutations. So what implications may they have for treatment, which drugs are likely to work, which drugs are unlikely to work. And then, of course, there's another step here in bringing these results actually into the clinics. So the way we're doing this now is to generate a report that's brought into the tumor board. And there's actually bioinformaticians sitting in the tumor board. And actually one or two of them are around here at the conference. They may be sitting in the audience somewhere. So I want to talk about these two steps a little bit in the next 20 minutes or so, mutation calling and some ideas about interpreting mutations. So I want to start with mutation calling. This turns out to be a very difficult problem for tumors, mainly because tumors are composed of a number of different cells that have a common evolutionary history, but they are not identical, and therefore there tends to be a large number of mutations that occurs at low frequencies. And although mutation calling has improved, of course, over the last past years, it is still very difficult to call low-frequency mutations. So depending a bit what you care about, but if you really want to do this, don't do very low-frequency mutations, it can be very challenging. So this is from some simulation that I don't want to get into here at all, but it does include low-frequency mutations, of course, looking at some common mutation callers, including those that you probably know, Mutate, VarScan, and so on, and some others. One of these is our own one. You see that the overlap in these calls can be very, very low at some point. So again, this remains a challenge. And one thing we did here is we did a little simulation study in order to assess where these differences come from and to get some idea of how it may be improved. We simulated a tumor by assuming such a hierarchy of the clones, the evolutionary history of the tumor, where the common ancestor cell or the normal cell is on the root of the tree here, and then we assumed some notion of this tumor evolving. And if you look at the tips of the tree, at the leaves of the tree, they would correspond to the existing subclones at the point in time where you sample the tumor. And these clones can have different frequencies, and again, some, and in general, probably many of them will have very low frequencies. So there's some frequency distribution over these clones. I will not go into all the details of the simulation, but the important thing is it's not a uniform population of cells. There are different subclones, and they occur at different frequencies. So then if you're willing to assume this sort of in silico generated tumor, mixture of tumor cells, you can generate reads you have to first place mutations into these clones. And we took these mutations from a real tumor, of course called from a real tumor. We placed them onto these different clones, then generated sequencing data from it, then processed the sequencing data, mapping, aligning, and so on. And then we applied different mutation calling tools. And because in the simulation setting we know the ground truth, of course, we can assess the errors, we can assess the performance, and we can also look at the reasons why certain mutation calls are wrong. Good, and the results are already here. So here we're looking at the sensitivity of finding a mutation as a function of the frequency of that mutation in the cell population. And this is just for two different false discovery cutoffs at 5 or 10%. And these are all the different methods. And as you see, if you go to higher allele frequencies, there's not so much difference anymore between the different methods. They're all doing pretty good, and this is what you would expect. But if you go to lower frequencies of these alleles, of these mutations, then there's huge differences, right? And some tools perform much better than others. So this doesn't work. So a second analysis is here. So here the performance has been assessed as the area under the precision recall curve and is plotted here as a function of the coverage. The coverage in principle also plays a role, of course. But, you know, in a reasonable range here, already beyond 50x, we went up to 200x. The differences are actually not so large anymore between different coverages. Of course, there are still differences between the different methods. So it turns out that the methods that tend to perform best in this particular comparison, the blue and the red one here, these are two models that first of all have a quite elaborate statistical model underlying the method, and second, they model explicitly the tumor normal comparison. The others also take that into account, but these are the only full probabilistic models, indicating that there may be some benefit in doing that. So again, because this is a simulation study, we know the ground truth, and we can also look now, and this is what we've tried to do here, for the reasons for the wrong calls. And so on the left here, we're looking at mutations that occurred at relatively high frequencies in the tumor, but have not been detected, so false negatives. So the question is, why were we not able to call these mutations? That didn't seem to be too difficult. And the two main reasons, the blue and the orange bar here, are simply low coverage, either in the normal sample or on the tumor sample. So it's the obvious thing, maybe. Coverage is, of course, not uniform, and in regions where you have too low coverage, we were not able to call these mutations. On the right-hand side are the high confidence false positives. So these are things that have been called with high confidence but have been false positives. And the main reason here is that these mutations also occurred in the normal sample, have been called there. Then we went on and tried to combine several callers with the idea that you may gain something from each of them, and maybe you are able to improve the performance overall in this manner. So again, this is as a function of the frequency here, showing the sensitivity. And we tried intersecting the calls from different tools. And we developed a way to combine the ranks of these tools. I will not show you the details of this method, but the basic idea there is to account for the correlations among the different tools. Obviously, all of these calls are not independent. In fact, they're highly dependent. They're essentially all calling more or less the same. And if you account for the dependency structure, it turns out you can do quite a bit better in combining the ranks here. Another thing I just want to point out, we also try to, for some of the tools, we try to optimize the parameters. Of course, all of these tools have several parameters, and the performance depends on that. Our analysis has not been exhaustive here in no way, but, you know, we try to explore for some of the, let's say, promising tools to move away from the default parameters, which was our default also, of course. And some of the parameters for some of the tools made a big difference. Good. So I want to point out that we developed a pipeline for the sequencing data, the whole exome, whole genome, and RNA sequencing data called NGS Pipe. This is a workflow based on SnakeMake, which is a workflow manager, basically. So this allows you to formulate the analysis of these sequencing data in a way that's very flexible, that is transparent. You know exactly what you're doing, relatively easy to use. And the idea is, of course, that this allows you to do these analysis in a very reproducible manner. As I said, there are predefined workflows for these three types of data at this point of time. And it facilitates the quality control, the documentation, and it also supports computations in a cluster environment, so you can do large amounts of data in a parallel fashion. Good. I would like to move on then to the second part of interpreting mutations. And the basic problem here, as most of you know, of course, is, well, in cancer, we would like to distinguish drivers from passengers. So we would like to detect mutations that are actually responsible for driving the tumor, maybe also for conferring resistance to treatment from the passenger mutations, which are just neutral mutations. Now, the problem is that most of the mutations are passenger mutations. Most of them are neutral, so you're looking a bit for the needle in the haystack. But also among the drivers, the problem is that most of the drivers are rare in each individual tumor. So you find maybe 3 to 5 driver mutations in a single tumor, but this set of 3 or 5 mutations, driver mutations, will be quite different between different individuals. And that makes the whole problem of finding these drivers very complicated and very difficult. So this is just a histogram, and you see there's a few genes that are quite common, and those are the ones we know very well, TP53 and so on. But then there is this long tail or heavy tail in the distribution, and all of these, and the tail is probably much longer, are very difficult to find. Now, in terms of interpreting mutations, there's many ways to do that, and many general tools have been mentioned in this course here today already, of course. And I want to start by talking about single mutations. If you want to interpret a single mutation, well, there's, of course, statistical approaches mainly based on recurrence. If a mutation occurs often, independently, in different patients, that's a strong indication, but it doesn't work very well, as I showed on the previous slide. Then you can try to do all sorts of functional predictions, maybe based on the structure of a protein, on orthology, on many other considerations. And I should say, for what's really used in the clinical setting right now is mainly based on curated data. And for example, we use this drug-gene interaction database that you can query, which is based on curated such interactions, which again come from different databases. So this is for single mutations, but I want to move on now and talk about interpreting mutational patterns. So each tumor will typically have a number of genetic alterations, and the question is, how can we interpret all of them as a pattern rather than independently of each other? So I think this is a nice figure from Ben Ruffel's group showing that there is a range of methods and you can sort of arrange them according to how much prior knowledge you use. And on the left-hand side, you would have things that are based on known pathways. So pathway enrichment analysis is the classical thing. Somewhere in the middle are methods that make use of interaction networks of one or another type. And on the right-hand side would be methods that make no assumptions, don't use any prior knowledge. They are typically based, of course, on finding patterns in the data. So they are statistical models, machine learning type of models. At the same time, the number of hypotheses that you're testing grows to the right, and on the right-hand side, these tend to be very hard statistical and computational problems, whereas on the left-hand side, you know, pathway enrichment analysis is something we pretty much know how to do. So there's many methods all over the place, and I just want to mention 2 or 3 that we have developed in my lab. So this one here is called Netix, which is based on an interaction network, and I will talk more about it today. We also worked on the right-hand side here quite a bit, so this typically boils down to finding patterns of co-occurrence or patterns of mutual exclusivity in the data. And we worked in particular on this problem of finding mutual exclusivity quite a bit and have actually 3 different methods of doing that. But I will not talk about it today anymore, but if you're interested, please talk to me. So I will focus then on the middle grounds here, using interactions, and I want to talk about this NetX method. So we're using network diffusion in order to propagate what you find from the primary genetic alterations. And as you know, the basic idea of this is kind of simple. So you have such a network, maybe a protein-protein interaction network, and you map onto this network the genetic alterations that you find. For example, this gene might have been mutated and all the others not. And the idea is then to say that, well, maybe the neighboring genes in the network, they also play a role in the process. That's the basic idea. Guild by association is the prototypical way of approaching this. And there's different ways of doing this in the context of networks. And this network diffusion approach is one, I think, elegant way of addressing this. So the way we like to formulate network diffusion is as a random walk with restarts, and essentially you have to invert matrices at the end of the day, so computationally this is all doable. So the method as a whole looks now like this. This is a bit of a busy slide, but I want to talk you briefly through it. So let's look at one sample here first of all. So first of all, we're using a directed network, unlike on the previous slide. And then we're doing two such directed graph diffusion processes. The first one starts with a number of genetic alterations. In fact, those can also be methylation events, or it can be events of microRNAs being differentially expressed. So we think of these as the causal events. And these we will diffuse in the direction of the directed network. And that gives you a diffuse distribution of the scores you start with. The second diffusion process starts with the differentially expressed genes, the blue ones down here. And here we're diffusing against the direction of the network, so exactly in the other direction. And the idea is then to combine the two diffusion scores from the bidirectional diffusion process and to find nodes in the network that get a combined high score. So where I get a lot of diffusion from the potential causes and I get a lot of scores diffused from the potential consequences. And we think of the consequence as a change in the gene expression program, in the gene expression patterns. And these genes are shown here in green, and we like to call them mediator genes because they mediate the effect of genetic and maybe other alterations, epigenetic alterations, on gene expression. So that's the basic idea, and we run these two, or this bidirectional diffusion process in each sample separately, in each sample individually. So it's done in this sample, in the second one, and so on, and so it's done in all of these samples. And we are ranking the genes then based on the combined score, so combined on how mediator-like they look like in this process. And again, we have this ranking for each sample individually, and at the end of the day, if you want, and typically you do want, you can combine these scores, you can combine these rankings into a global gene list. So first of all, we looked a bit at the performance simply by taking known cancer driver genes from different databases. I don't want to go a lot into the details, and we combined two methods that are somehow closely related, especially TIDAI also does a bidirected diffusion process, but not on individual samples. HotNet does a similar type of diffusion process, and DriverNet is a different method. And you see that especially for these, I don't want to talk about pan-cancer too much, but we're looking here at breast and lung cancer. We tend to recover these known cancer genes somewhat better. We also looked at the stability, so how stable is the final gene list under subsampling. And it turns out that the two diffusion methods here are much more stable, with NetX having some advantage again over TIDAI. So next, we did some enrichment analysis in order to see what do we find actually in this manner. And again, we did the enrichment analysis on the per sample level. So for each patient, for each tumor separately, again, we have a gene list. So for each of these gene lists, You do a gene set enrichment analysis, and you find a set of pathways, if any, that are enriched for this particular patient. And what I'm showing you here is the number of patients in which we find enriched pathways of a certain size. So the pathways are here ordered by size. And we're comparing this for the mediator genes, for the genes that we identify with our method, compared to just the mutated genes. So if you only look at mutations, and also for comparison to random genes. And you see that we find much more enriched pathways looking at these mediator genes as compared to the mutated genes. So the signal somehow, at least in terms of these pathways, and we did this for, I guess, reactome pathways and pathway comments down here. So the signal is much stronger when you look at these mediator genes as compared to just the mutated genes. Just one example here, this again for pan cancer, and we just picked out the number of pathways that are known to be associated with cancer, signaling pathways, Wind, TP53, and so on. And again, the mediator genes give a much stronger signal here as compared to the mutated genes. I want to quickly show you two examples. This is the well-known tumor suppressor gene TP53, also the most commonly mutated ones. So it also comes up, unsurprisingly maybe, as a mediator gene here. But the benefit of the method now is that you're finding this indeed mutated about in a third or fourth of the samples, but we find it downregulated, shown in blue here, in a much larger number of samples. And the model now predicts a number of different causes for this gene, this tumor suppressor gene, being downregulated. And this includes other genes upstream being mutated, being hypermethylated, or microRNAs that downregulate, that are known to downregulate TP53. So the idea is that these diverse mutational patterns that occur between the different patients all converge to the same consequence, namely TP53 being downregulated, and as a consequence, a whole part of the gene expression program being altered. So the way we see this method is really as explaining the mutational heterogeneity between patients and how they converge to the same or to similar phenotypes in terms of gene expression. Just a second example here that I picked, EP300, that I picked because this is actually never mutated. It's downregulated in a large number of tumors, but never mutated itself. Nevertheless, for us, it is sort of a key gene of key importance, although it's never mutated itself. And again, there's different causes here that we predict for it, including, again, microRNAs, mutations, of course, and methylation events. So with that, I would like to wrap up. So first part, I talked about mutation calling, and the starting point, again, was the molecular tumor board. It requires robust, transparent, and scalable computational tools for mutation calling and interpretation. NGS pipe supports reproducible analysis of these sequencing data, at least up to mutation calling. Nevertheless, calling low-frequency mutation remains a major challenge, and NATIX integrates different tumor omics profiles, predicts how the same pathway is affected in different ways in different patients, and it can be used to prioritize these mediator genes. With that, I would like to thank especially Christos, who worked on NATIX, and Ariane, who did the comparison of the mutation callers to PhD students in my lab. And thank you for listening.
I think we have time for a couple of questions. Did you consider individual subtypes in breast cancer, for example? Because P53 would be a driver in triple-negative breast cancer, as far as I know.
Yeah, no, we did not. So here so far we did the pan-cancer analysis where we're not so happy with, because exactly in your direction with the pan-cancer we're not so happy because we think for the same reason it doesn't make so much sense maybe. So we think we should look at the cancer types, but it's a good point to go a step further to look at the subtypes, but we have not done it yet. That's a good point.
How do you deal with hubs being enriched for mediators? So
How do we
hubs
deal
in a
with
network,
hubs?
highly connected nodes in a network.
We don't treat them in any specific way, but I guess you're referring to the diffusion process where they can, let's say, introduce effects that you may not like. Is that what you're referring to?
Yeah, so I would imagine that highly connected nodes in whatever network you're using are going to be more likely to be called mediators, right?
I didn't check it. I don't know. So I cannot tell you. We didn't check it.
So in addition to looking at the network-based method to prioritize cancer-driven genes, what do you think about to look at genes that are associated with PTM or domains in proteins?
So looking at genes that are
associated with PTM, like post-translational modification areas, sites and domains in proteins.
Yeah, I mean, sure, makes sense. I mean, one thing I didn't maybe mention, we did, in fact, for the breast cancer, not only look at gene expression changes downstream, but also at protein expression changes. So for breast cancer, there's quite a bit of protein data, also TCGA. But phosphorylation events we did not take into account because we didn't have any data on it. In principle, I do think they will be informative. So I think it's a good idea, but we didn't have the data here at this point.
Okay, thanks.
I think we have time for the last two questions.
Thank you again for the talk. Maybe you already mentioned it, but what network do you start with? Do you calculate it initially from the data, or are you taking a known network into account?
Thanks. That's a good question. I did not say it. So it's a network we took from a publication where people have integrated different data sources. So it's a curated network. I mean, it's based on CAG and on several other sources. And again, it's a directed network, so that's very important for our approach. Yeah.
So